Welcome to lesson 6.7. This lesson focuses on soil protist symbioses, and we will be discussing in detail the cellular slime molds. These organisms are by far some of the most fascinating organisms on Earth and some of the most widely studied. So I really hope you enjoy this lesson on cellular slime molds. So one of the best examples, perhaps, of how soil protists work together in cooperation in a symbiotic relationship is that of cellular slime molds, where individual amoebae that normally live on their own under stressful situations such as starvation, food scarcity, start to cooperate and become one organism. There are two main orders or groups of organisms that are classed as cellular slime molds, and that's the Dictyostelidae. And there's meant to be a second I in here, so just correcting that spelling mistake there. But these are orders, but basically you can look at them as two different groups of unrelated organisms that form cellular slime molds, and we'll actually concentrate on the Dictyostelids, so the Dictyostelida order. And the most studied genus of Dictyostelids is Dictyostelium. So there are obviously different species, but this is one of the most widely studied example of this relationship between individual cells' ability to cooperate in such a manner, coming together to form a multicellular organism, you could say. So this is what the scientific world currently believes, that multicellular life, such as us, for example, occurred as a result of this cooperative behaviour of individual cells. And then these cells began to differentiate, and they actually do this differentiation in the cellular slime molds as well. So let's just look at how this occurs, and then we'll go through the life cycle. So single-celled amoebae produce a chemical signal called cyclic AMP when they are stressed, and this causes an attraction via the process of chemotaxis, which is recognition of a particular chemical signal in the environment. They start to swarm, they come together, and they form this structure called the pseudoplasmodium, or another word for that is a multicellular slug, or just slug. First of all, the amoebae just feed on bacteria, they go about their own business, doing the individual life thing, doing the single life thing, and multiplying. So going through binary fission, each individual amoeba making a replica of itself, making its own clones. And as a result, there is a mass of amoebae feeding on the bacteria, particularly in that particular environment. And then all of a sudden, bacteria run out. And this could be because there are more amoebae replicating and feeding versus how quickly the bacterial populations can recover. Or it could just be that the environment becomes dry and bacteria are no longer multiplying, or that the bacteria have run out of food themselves. There's no more organic matter on which the bacteria are feeding, and as they have run out of their food source, and so the amoebae run out of a food source. There could be multiple reasons as to changes in the environmental conditions causing starvation for the amoebae. So they become aware that starvation is a potential reality for them, and they then go through this chemotactic aggregation towards the signal that these stressed cells are putting out. This chemical release and this aggregation allows the individual maybe to start creating a multicellular mound. And this produces a finger where the mound actually stands up in such a fashion that it produces something that's been likened to a finger, and then the motile pseudopodium is formed, called the slug. Then the slug itself, as it moves along, finds a suitable place and produces a fruiting body, where at the top there is a spore mass that forms, and in this case it's called a sorus, and some cells die in order to produce the stalk. So very similar to the acellular or the plasmodial slime molds, but different in the sense of that now you have this swarm of many cells coming together to form the slug, versus, remember, in the plasmodial slime molds, it was two cells mating to produce one big cell. In this case, these cells remain individuals and act in unison as one organism called the slug. When the food source once again becomes available, the spores germinate and the life cycle begins again. So if we were to look at it on the diagram, we have the growth phase. So that's amoebae just doing their thing individually and going through binary fission as an asexual way to reproduce. And as more and more amoebae develop, eventually they run out of their food source and starvation starts to occur. And this causes the aggregation signal, the cyclic AMP, to be released. And this allows the cells to come together and start forming what is called the aggregation stream. So this is, you can imagine all the amoebae coming together to this one spot, and then that creates a mound, and there's a tip that grows, so allows the amoebae to climb up, and eventually a finger is formed, then it falls down, usually to move. And eventually this slug structure stops and it goes to this culminating point, looks like a Mexican hat, and then a stalk is formed from this. So these cells will die, and then the reproducing cells will form spores, and the spores disperse. So this is the sorus. So let's just look at another example of this, but also to demonstrate the insane capability of these organisms to develop an immune system while they're in this aggregated slug form. So Dictyostelium discoideum goes through that exact life cycle. This diagram also involves making a point of the extracellular matrix, and this becomes important in this immunity mechanism that this organism has. So let's just go through it here as well. Once the spores germinate, they go through their growth phase, so they multiply by binary fission. Lots of cells are formed as they replicate, and then starvation happens, they run out of food, and they start aggregating, start the aggregation process, they get together through this cyclic AMP, and then the mound is formed with this now extracellular matrix. This standing slug forms, which is also called the finger, and then when it falls down, it starts to enter the culmination stage, and this is where the stalk is formed from the Mexican hat, and the sorus starts to develop, and of course that grows and eventually it matures, and the fruiting body or the sorus releases the spores. So basically, if the slug is already formed and it's underground, it migrates to soil surface because that allows it to disperse its spores, and this rudimentary immune system begins to form. What's really interesting is that what it forms is specific cells called sentinel cells. The word sentinel or sentinel cells describes any cell type that plays a prominent role in host defense by being aware and sensitive, designed to sense and monitor the presence of any injurious substances, materials, or organisms, and to alert the rest of the cells or the rest of the body to their presence so that an immune response can be launched. So just like our bodies have sentinel cells, so do these slugs. These amoebae actually form similar cells in order to defend the slug, the entire multicellular organism that has been formed by the individual cells. So it's a very beautiful process that these organisms go through, and it's very similar to that of our own body. And these are phagocytic, so they basically engulf anything that might attach to or be harmful to this migrating slug. So like any bacteria that might want to eat it, these cells actually start to attack any potential threat. And they actually move throughout the slug, and you can see here in the diagram, we've got the sentinel cells, and they basically travel through, and the extracellular matrix leaves a thread of these sentinel cells to protect the slug, and the protective sentinel cells are shed through this extracellular matrix. And this is what these cells look like. They basically form this extracellular trap, which is the ET part. It's not extraterrestrial, it's extracellular trap. And the bacteria get engulfed in that. And that trap is formed by the mitochondrion. And there's a lot of immune system-like traits in these sentinel cells. And one of the chemicals involved in the immune system, both in humans and obviously a lot of different organisms, including these unicellular amoebae, is NOx, NADPH oxidase. Basically, NOx allows for reactive oxygen species formation, and these are once again molecules that play a major role in cell survival, death, differentiation, signaling, and inflammation. So very much immune response like signaling going on. And then also interestingly, there is this tier A protein that contains interleukin receptor domain, and interleukin is important in immune cell activation, differentiation, proliferation, maturation, migration, and adhesion. So basically the reason why I've put all this complex stuff in here is not to bog you down with unnecessary information, but to show you the complexity and the marvelousness of demonstrating how individual cells, when they cooperate together, can form this little living being. And they're still individual cells, they can still walk away at any point, but they create together this incredible structure that then has its own immune system and its own sentinel cells that are actually protecting them, just like we have our different sentinel cells that protect us and defend us from bacterial attack and toxins, and they actually go around and consume anything that's a threat to our body. And this is actually happening in the slug. So that to me just feels extremely fascinating and extremely important to understand the complexity of this microbial world, and even more so, what happens when organisms cooperate. What would happen if humans actually cooperated? What could we achieve on Earth as a result of this cooperation? There's a lot of analogies that can be drawn. And of course, scientists are looking at all this and studying this very carefully to understand these processes of how single-celled organisms, when cooperating symbiotically to form a multicellular cohesive structure, the extreme benefits that occur as a result of that cooperation, as a result of that symbiosis. So this is borrowed from a YouTube video called Dictyostelium, a cellular slime mold by Jeremy Pickett Heaps. So you can look that up on YouTube. I am going to show you snippets out of this video as to the actual stages, how amoebae create the multicellular slug. And so first of all, we have the amoebae feeding on the bacteria. And once the food supply becomes limited, the amoebae basically start to swarm. So they follow this cyclic AMP release that's being triggered by the starvation of the amoebae population. So the cyclic AMP is like a hormone that attracts amoebae from all different directions. And they start to swarm, moving forward to the aggregation center where they're all going to meet. And you can see here the swarming behavior. And they synchronize in order to conglomerate into one multicellular unit. So once they accumulate into these aggregation centers, there are going to be many separate units of these aggregates. It's not just one giant structure that's being formed, but it's several pseudoplasmodia or several slugs. Because remember, there's masses, millions of these cells coming together, and there's a critical mass as to how many cells can collectively make up a functional slug. So sometimes the slugs can actually fuse to further increase their cooperative behavior. So the two will merge and they will join forces in looking for the right place to land. And the slug is made so that collectively, the amoebae can decide where is the best place to stop in order for the future generation, once the spores are formed, to be released and land in a suitable area, a suitable substrate for them to feed from. So once a suitable place is discovered, the slugs actually conglomerate in that area and start the process of forming the stalk, fruiting body, and the sorus to disperse the spores. Even though the amoebae remain as individuals within the whole process, there's actually some cellular differentiation. Not only do some form the sentinel cells that defend the slug, there's also this division in labor in terms of who goes into the stalk and who goes into the fruiting body, giving rise to the next generation of amoebae. So it's a very organized process that comes from individuals creating the future for that species. So as a result of this incredible potential that these organisms display, social development studies have been undertaken, and in particular studying unicellular altruism. Before I describe that, I just wanted to show you this image on the left that goes through the different 24-hour cycle that these organisms organize themselves into during that time in order to sporulate and avoid starvation. So the first thing that happens is once the aggregation stage occurs, there's this loose aggregate that forms within 10 hours. Within 12 hours, this becomes a tighter aggregate, and at 14 hours, it starts to produce the tip. Between 14 and 16 hours, it migrates as a slug to a suitable position. At 16 hours, the finger's formed, and then the Mexican hat is formed. I love the names that have been given to all these structures. And between 20 and 22 hours, there's the early culminants. Then the stalk starts to form here, and the fruiting body develops by 24 hours. And then, of course, at the end, we've got the sorus that's going to split open and release the spores, and the cycle will begin again, where the free-living amoebae will live out their life until such time as conditions are right for them to get together again. But what's really interesting is that when scientists are studying these organisms, they're noticing that different clones, because remember the amoebae are individual clones, and the population will be made up of various clones of one particular amoeba and another particular amoeba. So there are differences between individual amoebae that made their own clans through the clones that they've replicated from themselves, so that are genetically identical to them. And there is a difference between clones that end up in the stalk and those that end up in the sorus. And remember that in the stalk, the cells are actually sacrificed. They will die. So about 80% of the cell population will end up in the sorus and will actually survive to reproduction. And then 20% of the population of cells end up in the stalk cells, and that means imminent death. And there are actually cheetah clones. So these are particular amoebae, particular clones, where they disproportionately outnumber other clones. So there's a disproportionate number of those particular cheetah clones in that sorus. So obviously, scientists are looking at it from the perspective that these guys are onto something, like, hey, let's survive. We're going to put more of ourselves there and let the other clones take the slack and self-sacrifice. And this is something that's been noticed, that while some of these amoebae clones are altruistic, others actually take advantage of that altruism and disproportionately are driven to become spores. We don't know what the mechanics behind that is. Perhaps there's certain genes in those that are more favorable in particular conditions. We don't know, and I'm just speculating here, so this is not something that's been studied or I'm aware of, but I'm speculating that maybe there is some other mechanism that drives this and they're not just cheats. But there's another very interesting example of this cooperative way of living, in that 1/3 of the population become farmer cells, where they actually harness bacteria farm bacteria. So bacterial husbandry is happening, where they store the bacteria and they don't consume them, but store them for the release with spores in an area that is lacking food. So this is particularly strong where the amoebae know that food is going to be lacking, so they prepare a little food packet of bacteria for the germinating spores. However, the rest of the population of the amoebae, so the 2/3, are non-farmers, but their progeny benefits if they end up in the sorus. So the farmer clones additionally stored unconsumable bacteria such as Burkholderia and Pseudomonas, and these serve to produce molecules that enhance the farmer clone's spore production. So if some of the farmer clones get into the sorus, they will have the advantage by having the bacteria to encourage or enhance their spore production while suppressing the non-farmer's spore production. So while this might sound altruistic to begin with, there's also an advantage, and these organisms obviously have a distinction between themselves, their own clones, and those of other cells. So a very interesting scenario, and I'm not sure exactly how this works and how much of what we're seeing is driven by humans observing this process. So while this behavior in slime molds is quite fascinating, I would like to stress out that these cell lines have been grown in the lab, and some of them have been grown for decades, where the lines have been used over and over again. Another thing is that when these experiments are carried out, there are often only 1 or 2 different clones used. There's such lack of biodiversity in these systems when scientists study these relationships in the lab. So we don't know whether these phenomena we've just described and the attribution of altruism in social development studies is actually happening in nature, given that there might be many more clones existing in nature. We don't know really what is happening in terms of the reality of these relationships in nature. And so my worry is that we attribute all these human characteristics as these specific examples show, and it just makes me wonder whether these attributes are in fact a phenomenon that occurs in nature, in biodiverse systems, and at the same time giving it human-based characteristics or human-based behavior is also something that I am not entirely sure is an accurate description of what is happening. Also, the word altruism suggests a feeling of compassion, which I am 100% sure these organisms don't have. So it's not driven by their personal desire to be altruistic towards others. However, there is a possibility that the design of these creatures, or any other creature for that matter, is such that the inbuilt ways in which these organisms work can be interpreted as altruism, just because nature is designed to be giving and be sharing and be productive and work for the greater good. But that says more about the creator of the system rather than the actual creatures that are non-human. As we know, humans are the only ones that can actually have feelings that generate altruistic behavior or any other beneficial social behavior, or otherwise, or contrary behavior that is non-altruistic, that is negative towards others. So I just wanted to put my opinion in on that, just because I don't always agree with the way science is interpreted or extrapolated, and how often we use nature to explain our own behavior. We have to take way more responsibility for our behavior. So while this is all fascinating, I have some questions about these scientific experiments and whether what is actually being explained here is the truth or the reality of what actually happens. So I just wanted to say that because I think there's much more to it, and science is not flawless. But very interesting that science is looking at these and discovering these different modes of social behavior and social development in slime molds. So as a result of these incredible organisms doing what they do, i.e. getting together to form social structures getting together to form multicellular organisms. Scientists study these guys a lot, and they are in fact the model organisms for biomedical and human research because they show so many similar characteristics to those of human cells that it's easy to have these model organisms and study particular phenomena in biomedical science. They also display very fundamental cellular processes that all cells have. It makes them a perfect model to study these processes. Another reason why these organisms are very useful in studying human and other cellular processes is that they exhibit a lot of conserved cellular and developmental processes, and this means that they are conserved across species. So genes that are conserved or cellular processes that are identical across different species of organisms. In fact, different groups of organisms such as plant cells, animal cells, fungal cells. So this is why slime molds are so good to investigate, because they do share that in common with other groups of organisms. There are also great models for studying host pathogen interactions, as you have seen with bacteria that might want to eat the slug. And also various nervous system diseases can be studied using these organisms. So there's just a plethora of various ways these organisms can be used, and science is using them in order to investigate a lot of phenomena, both biomedically and otherwise. The third group of slime molds are called protostellids, and these guys are also made up of amoebae, but they produce simple fruiting bodies, including a stalk with one or few spores. So they're much simpler and they're not well studied because they're all pretty much microscopic. There is also no model organism that is being used in studies, so very little is known about protostellids. But they dwell on dead plant matter, so they help in nutrient cycling, breaking down organic matter. Like other slime molds, they consume bacteria, yeast, and fungal spores. And this is just an example to show what these protostellids look like. This is the amoeboid stage, and this is the fruiting body stage. So very similar, but much simpler, and not many are discovered because you can't see them. They are not easily observable in nature, so they are not as widely isolated as the other slime molds. So this is pretty much it for the slime molds, and the purpose of having gone through these two groups of slime molds particularly, I really just wanted to demonstrate just the complexity, the beauty, and the wonder that is in these humble protists that live in soil in the two various forms, the amoebae and the flagellates. But in particular, this lesson being about cooperation and symbiosis, I think the cellular slime molds perfectly demonstrate the benefits of having symbiotic relationships and cooperation with one another. So we have come to the end of lesson 6.7 on cellular slime molds. And in the next lesson, we'll continue on the topic of soil-protist symbiosis, looking at mixotrophs, parasites, and macrobiotic crusts. So you'll be hearing from me again in lesson 6.8.